Hello and welcome to part 3 of this introductory lecture on the nervous system. In parts 1 and 2 we looked at the central nervous system and the peripheral nervous system, and on this part, in this segment of the lecture, we're going to look at some supporting cells and a couple of other things to finish out this lecture. So here we'll look at what are called neuroglia or neuroglia. It's also pronounced that way, or just simply glia. Now, neuroglia literally means nerve glue. Okay, so and they kind of do that. They do hold things together. They're like the supporting cast for the neurons. So these are the guys that are the cells that are helping out the main actors, which of course are the neurons, and these are the supporting cast for them. They have 4 general functions. Now each one has its own specialized structure and function, but overall, if we wanted to look at the functions of the glial cells, we could say that I tried to narrow it down to 4 general functions. To surround neurons and hold them in place, to supply nutrients and oxygen to the neurons, to separate one neuron from another, to destroy pathogens, and remove dead neurons. So overall, this is kind of what they're doing, but each one, like I said, has its own specialized structure and function. And so without them, just like if you had, in a movie, you've got the main characters. Without the supporting characters, I mean, you don't have a movie. I mean, it wouldn't make sense. So with the nervous system, you've got the neurons, but without the glial cells, the neurons are in a lot of trouble. Things aren't going to work at all. So because glial cells divide, and neurons generally don't divide, most brain tumors are gliomas. And if a word ends in oma, it means it's a growth or a tumor or a mass of cells. So most brain tumors are gliomas. There are 2 glial cells in the PNS, and there are 4 in the CNS. And then these 2 video links here, glioma excision, you can see that. Go to YouTube and type that in. I just found one that was pretty decent. The video quality was really good, and the patient had a glioma in his frontal lobe, and the neurosurgeon just removed it. And you look at it, and what I want my students to see whenever they watch this video is the texture of the brain and how difficult neurosurgery is, because the brain, it's not like you would be cutting into connective tissue or muscular tissue where there's kind of a solid substance to the tissue. Brain tissue is not that way at all, and this video shows how that is the case. The glioma is removed, and the patient, it was a successful surgery, and they show the patient post-operative, and he's doing well. Then there's also a very cool thing, and a lot of this gets loads of views on YouTube, awake brain surgery. So go to YouTube and type that in. This one here in particular that I chose, which I thought was cool, this was an opera singer who had a benign glioma, and he was having it removed. I think that's what it was. And while he was having his brain surgery done, he was awake, and he starts singing. He's singing in Italian, and he's singing some opera portion of an opera, and the neurosurgeon must have hit some part of his ability to sing and to speak, and his singing goes awry, and it sounds kind of funny. And he was totally aware it was happening. It looks like he was a little scared, but there's a great nurse by his side, and she was comforting him and telling him he'd be okay, and he was doing great. There's others where a guy's playing a guitar while he's having brain surgery done as well, so it's kind of a cool thing to watch. So if you want to check that out, go ahead, type in awake brain surgery. So we'll look at the 2 PNS neuroglia or glia. Schwann cells. And Schwann cells are named after a scientist. His name was Theodore Schwann. He was involved in, if you have biology, he was involved in the devising of the cell theory. He discovered and stated that all animals are made of cells, so this went against spontaneous generation, and so he helped form the cell theory. But he was very much involved in the nervous system, and he discovered these cells, and so we named them after him. We call them Schwann cells. They form the insulative myelin sheath around the axons. like the covering around electrical wires that keep the electrical impulses within the axon. They enable quick conduction of electrical impulses within an axon. And so if you look at the picture down below there, the yellow portion of the structure, that's the axon. So imagine the axon, imagine this is like an electrical wire and it's been cut in half, longitudinally, like right in half horizontally there. And what you would be doing is looking inside the axon. And then the axon is the yellow part. And then the green, the green layering, that is one Schwann cell. And it wraps itself around and around and around multiple times around the axon. And the more layers that's wrapped around it, the more the insulating ability of the Schwann cell. And if you look at the little axon in the middle, you can see on the right-hand side of it, there's this bump, this green bump on the right-hand side. That would be the cell body. That's where the nucleus and the organelles would be. And then from that, so you would, if you begin at the cell body and then go down, that would be the extension of the cell membrane wrapping around and around and around in a clockwise fashion. So that's the Schwann cell wrapped around the axon. The axon, again, is the yellow. The outer layer is called the neurolemma. The neurolemma. It's also pronounced neurolemma or neurolemma. It's spelled with an O or an I in it after N-E-U-R-I, neura or neurolemma. Protective and regenerative layer of the Schwann cell. And so that's the layer where if any damage is done, it's a regenerative type of cell, and it can repair itself and regenerate. And so these are the cells that form the covering around it. With multiple sclerosis, these are the cells that are damaged, and you develop scleroses or scars, and that's why it's called multiple sclerosis, because you have many. There's a lot of scarring that's forming because this is an autoimmune disease where your immune system attacks the Schwann cells, and then you can't conduct impulses properly because the Schwann cell is no longer wrapping around the axon. There's damage that has been done to it, and so electrical impulses do not get conducted properly as a result of the damage to the cells. Then the second type of a glial cell in the PNS is called the satellite cell. Satellite cells are protective, cushioning cells. They appear to be involved in the regulation of pain, and they connect neurons with the blood supply. So these guys are bringing in nutrients and oxygen to the neurons of the PNS because they're connecting the neurons with the blood supply. So they're a go-between between the circulation, the blood, and the neurons themselves. It appears that they're complex cells that carry out a wide variety of functions. There is still much to be learned about them. So notice I use the word appear here because we're not sure. There seems to be a lot more going on with satellite cells than we think. They're kind of there's some things about them and their functions are still a mystery. And so you can see in the picture how they wrap themselves around the neuron cell body. The satellite cells, see they're labeled there, and they're surrounding them, and then you can see the Schwann cells wrapping themselves around the axon. So these are the two of the PNS, the Schwann cell and the satellite cell. Now we'll look at ones, there are 4 in the CNS. Microglial cells. These would be nicknamed the cleanup crew, and they're phagocytic. They're activated after infection or disease, and they remove damaged cells or kill invading microbes or pathogens. So these ones are cleaning things up. They're getting rid of anything that may be threatening to it or anything that's been damaged. And they are derived from blood stem cells. So they begin in the red marrow as a blood stem cell, and then they undergo differentiation where they take on their own specific function, and part of their genome is activated, and they become and are assigned the role of a microglial cell in the central nervous system. And this is a micrograph, the quality is not the best. It's hard to get a picture of these guys. This is through fluorescence microscopy so that you can see the difference between the cells with the fluorescent dyes. The microglial cell is green after injury to a brain capillary. So this would be a capillary, part of the blood-brain barrier. Remember, we're in the central nervous system, so this is part of the blood-brain barrier. The microglial cell extensions surround the injured area, and I ask why? What's it doing? Well, look at its function. We just went over that. They're the cleanup crew. They're fixing damage, they're repairing damage, and they're getting rid of any pathogenic cell that may be present. So they're ensuring that things don't become infected. Second type would be the astrocyte. Astro meaning or referring to star, and cyte means cell. So these are star-shaped cells, sort of. They're the central nervous system equivalent to satellite cells in the PNS. Which means, well, sorry, I gave you a number of functions about satellite cells, but one of them was connecting the neurons to the blood supply. And that's what astrocytes are doing. They're the connecting cell, they're the intermediary between the blood and the neurons of the CNS. They're an integral part of the blood-brain barrier. And they're the most abundant cell in the brain. There are more astrocytes than any other type of cell in the brain. And in the picture on the left, the micrograph on the left, the astrocyte is kind of, well, you see where those blue arrows are pointing? That's where the astrocyte's legs are connected to the blood capillary. So that orange, that's a tube, I guess. You can't really tell it's a tube, but that orange, what you can imagine would be a tube, that's a brain capillary, and the astrocyte is connected to it there. And the astrocyte is that dark orange thing that kind of looks like a star with two legs coming out of it. And they're connecting the blood supply from there, which are where you can see connected to the brain capillary, and they're connecting that to the rest of the neurons. The picture on the right, the soma, that's just the Greek word for body. That's the cell body. That's where the organelles would be in the nucleus. And then they have their processes that come out, and somebody thought, hey, they look like a star, and so they called them astrocytes. So connecting the blood supply with the neurons, very important part of the blood-brain barrier. The third type of CNS glial cells is the Ependymocyte, and it lines the cavities of the brain, which are called ventricles. That's what should be in parentheses there. And they're the ventricles within the CNS. They assist in the production and circulation of CSF, or cerebrospinal fluid, with the help of hair-like cellular extensions called cilia. And so you can see in that micrograph there, the spinal cord, part of the ventricle that runs down the spinal cord there. You can see the ependymal cells are on the bottom there, and then they have these cilia extensions. And what the cilia do, cilia can be used for a variety of functions. In this case, it assists with the circulation of it, the movement of cerebrospinal fluid through the central canal, which is an extension of the brain ventricle running down the cord. And so that's their main function. The fourth one, the final one of the central nervous system glia, is the oligodendrocyte. This word is basically three Greek words in one. Olig means not many. Dender means branch-like extensions, is what that's referring to, and cyte is cell. So there aren't very many of them. They have branch-like extensions, and they're a cell. They're like Schwann cells. They form the myelin sheath around axons, but this is in the central nervous system. And one oligodendrocyte can form the myelin sheath around as many as 50 axons from 50 different neurons. Schwann cells, they can only form the myelin sheath around one axon. And so the picture on the left is a depiction of what we think it looks like, and then you can see the one on the right, the oligodendrocyte is massive. It's that big gray blob in the middle. And you can see the nucleus, which is a massive nucleus inside the cell. And then all of those circular, sort of circular things around it, those are all axons. And that oligodendrocyte has formed the myelin sheath around all of those axons that are surrounding it. And then there are the two video links there. They're somewhat related to this. They're both TED-Ed videos. Can the brain heal? And parts of it can. Some neurons are regenerative, many are not. And then myelin and imagination. And there's been discoveries found not all that long ago that myelin and the amount of myelin that's present, that's formed from these oligodendrocytes in the CNS, the amount of myelin seems to be related to the ability for imagination and brainstorming and coming up with novel ideas and unique ideas. That seems to be involved there. And myelin formation in the CNS begins early in our childhood. And so all of this, the reading that a young child can do and all of the pretend playing that they do, it is so important during that time period because it's leading to the production of more myelin around the CNS, and this makes nerve conduction more efficient, which makes thought processes happen more efficiently. Unfortunately, with all of the video gaming that's going on and all of the electronic amusements that we have, that has been found to reduce the amount of myelin that forms around the axons whenever young children are continually in front of a screen, because it's not allowing them to become imaginative. Imaginative play is more than just play. I mean, this is leading to a physiological development within the central nervous system. So this is a good video. It's kind of strange video, but it's a good TED Ed video that shows how myelin formation and imagination go hand in hand. And imagination then becomes, later it becomes logical processing thinking and intelligence and mathematical reasoning. So, we make that progression and that maturation as we get older. It's when we're kids, it seems cute, it's just imagination, but when we're adults, it's so much more serious. But that's when it all begins, is when we're children and our imagination. And allowing a child to imagine and pretend play is so important in their development. So important. There's so much you can say about that, but I've said enough. The nodes of Ranvier, named after obviously a French scientist. The nodes of Ranvier are gaps between the Schwann cells in the peripheral and oligodendrocytes in the central nervous system. They increase the conduction of an impulse down an axon. And what they do is they allow the impulse to, quote, jump down the axon, and that's called saltatory conduction. And it comes from a Latin word, that saltare, which means to jump or to hop. And so this enables quick conduction. So nodes of Ranvier are important in nerve impulse and the conduction of the impulse. Because the axonal membrane, or the membrane around the axon, is exposed at the node, nutrients and wastes are able to enter and exit the node. So the node kind of opens things up and allows waste to be released into, if it's the central nervous system, into the cerebrospinal fluid, if it's the Schwann cells in the PNS, into the circulation. And so these wastes can be removed, and because the axons are quite active, and with the myelin sheath around them, they cannot get rid of their wastes because the myelin sheath is pretty much impenetrable to those wastes. So these nodes of Ranvier are an opportunity for the waste to leave, and also for nutrients then to enter as well. So these are little gaps that form along the between the Schwann cells and the oligodendrocytes. Nodes of Ranvier, so they're important in terms of conduction and the health of the axon. And our final slide is, I just want to mention the synapse. And these are, the synapse is the region where one neuron meets but does not touch another neuron. So it's a gap in between them. And in this micrograph here that's been colored for us, the vesicles, which are the purple spheres, vesicles are basically membrane-bound sacs within the cell that carry something, and in this case they're filled with neurotransmitters in the nervous system. Of course, in other cells it can be filled with all kinds of other stuff, but in the nervous system, neurotransmitters. At the synapse, the vesicles fuse with the neuron's cell membrane and release their contents. So if you can look at this picture, you've got a neuron at the top, and then you can see the synapse, which is kind of dark and has that reddish coloring to it, and then there's a neuron down below. So the vesicles are traveling down through, they fuse with the membrane because they have a phospholipid bilayer around them, and so they can fuse with the cell membrane of the neuron and then release their contents, and then those neurotransmitters are picked up by receptors on the receiving neuron's cell membrane. So those receptors are waiting for the neurotransmitter. They get picked up and then passed on to the next neuron. And then there's a cool video about how neurotransmitters are transported. It's an interesting video. I suppose if you went on YouTube, I don't think neurotransmitter transport would be enough. Maybe that plus carrier proteins, something like that. And it's a pretty cool video about how neurotransmitters are. It's like a day in the life of a transport protein or something like that. But it has really cool animations. So that ends part 3 and the final part of the nervous system lecture. So hopefully you enjoyed this, learned something from it, and I hope that you share this video with others. If you know anybody else who's studying the nervous system, please share that with them. And as always, thank you for watching.